Hello, my name is Susan Northfield and I am the President of the Victorian branch of the Royal Australian Chemical Institute. The RACI is the official professional organisation for chemists across Australia, and our membership is vast and diverse. Among our many activities, we spend a lot of time working with schools to encourage and inspire the next generation of Australian scientists. Of those activities is our annual Hartung Youth Lecture. The Hartung Lecture is named after Professor Ernst Hartung, who was a well-known Victorian chemist at the University of Melbourne in the 1920s through to the 1950s. He was a prolific researcher and an enthusiastic teacher who was well known for including live demonstrations as part of his lectures. He was also president of the RACI from 1927 to 1933. To honour his contributions to Australian chemistry, each year we run a lecture series for Victorian and Tasmanian high school students. Applications are received from members of the RACI who wish to take on the title of Hartung Lecturer for the year. The lecturer then goes out to schools and gives a presentation about a field of chemistry that excites them and includes live demonstrations to inspire our budding next generation scientists. I am delighted during National Science Week to announce that our 2022 Hartung Youth Lecturer is Associate Professor Rajesh Ramanathan from RMIT University. In a moment you will hear from Rajesh, who will give an overview of what his lecture will be about. Afterward, I will let you know how you can arrange for Rajesh to come out on site and give his full presentation. And now, over to Rajesh.
Hello, my name is Dr. Rajesh Ramanathan, and I am an Associate Professor at RMIT University. It is a great pleasure to be awarded the 2022 RACI VIC branch Hartung Youth Lecturer. I will be visiting schools across Victoria and Tasmania over the next 6 months and will speak to you about what it is to be a scientist working in a chemistry laboratory. Specifically, I will be speaking about some of my research around detection technologies. Over the last 2 years, we've seen the impact of COVID on our lives. It has changed the way we do things. How does my work fit with COVID? Well, my research is about developing new technologies to detect viruses such as COVID. In fact, not just viruses, the technologies we develop in our lab can be used to detect a wide range of targets, from antibiotics in soil or animal products to gases in the environment or diseases in humans or animals. You might wonder how easy or difficult it is to make these technologies. What are some of the steps involved in developing a technology like this? And how does chemistry fit into all of this? My Hartung lecture will answer these questions. Let us imagine a situation that highlights why we need to develop detection technologies. Let us assume that you have a small cut on your leg. You see that this wound is getting infected. You decide to go to a doctor. The first thing they would do before treating you is to find out which bacteria is causing this infection. Without this information, it would be very difficult to treat the infection, as the same antibiotic cannot kill all bacteria. Let me show you a short video of one of the detection technologies we have developed to see how we can determine if a person has a bacterial infection, and that too quickly. You can see that we use a cotton swab to collect the sample. The sample collection plays an important role in developing a detection tool. Typically, the doctor would collect the sample and send this to the lab for analysis. What we want to do is to create a tool that the doctor can use on site to determine if the infection is caused by a bacterium. We do this by using a color-based detection system. For instance, we have 2 samples. One of them has no bacterial infection and the other has a bacterial infection. We start with 2 tubes showing a blue color solution in them. If the swab is positive for bacteria, it would change color, else it would remain blue. Let us see what happens when we put the swab into the solution. The first tube changes color from blue to yellow almost immediately. However, the second tube does not change color. This tells you that the sample in tube 1 has a bacterial infection and the second one does not. Based on this information, the doctor can start treating you straight away rather than waiting for the lab results to come in. This video shows you the importance of developing rapid detection technologies. During my talk, I will cover aspects of the underlying chemistry involved in the development of such detection tools. I will also talk about the working principle of the COVID rapid antigen test that I'm sure we've all seen before. Additionally, I will demonstrate how we can make such tools, where I'll show you how we make nanoparticles, how we can use nanoparticles to make such color-based detection technologies. I am looking forward to talking to all of you and using live demonstrations to show you the exciting field of analytical and materials chemistry.
Thank you, Rajesh, for a glimpse into the Hartung Lecture of 2022. If your school wants to invite Rajesh to give the full presentation to students, please use the expression of interest form in the description box below. And if you have any questions, you can reach us via email, Twitter, Facebook, or LinkedIn. All details for those accounts you can find below as well. For more information about the RACI, please visit our website. Happy National Science Week. Thank you for joining us.